Hello, everybody. Thank you for a nice introduction, and it's an honor to open this section. And since we are here in the Faculty of Law, so I will tell the truth, only truth, and nothing but the truth. So, and now we can start with how misplacing TDP-43 messes up with its protein network. So, I'm under oath, so I can tell you now the truth. So, and this is that we are getting older. Dear colleagues, we are getting older. And yes, as population continues to get older, we are moving closer and closer and closer to an epidemic of neurodegenerative diseases such as Parkinson's disease and Alzheimer's disease. But I'm not here to talk about Alzheimer's disease or Parkinson's disease, but about amyotrophic lateral sclerosis, or shorter ALS. Well, I know that you all know this disease, and you probably remember this ice bucket challenge, where many celebrities, and when I say many, I really mean many famous celebrities, decided to take part in this challenge to support patients and inform about this devastating disease, and of course to raise money. Well, so I know that you're not sclerotic. You already heard a lot about this disease, but just to refresh our memory, ALS is progressive neurodegenerative disease that is characterized by progressive degeneration of nerve cells in the spinal cord and in brain. So while research into ALS is advancing steadily, we are still lacking of efficient therapeutics and early diagnostic markers. And to make this problem even bigger, many mutations in various genes, such as FUS, C9 or 75, TDP-43, cause ALS. And now we will shift our focus on TDP-43, which is sometimes also referred as a fallen angel. And now let me tell you a story about this fallen angel. So TDP-43 is a DNA and RNA binding protein that is comprised of N-terminal region with nuclear localization signal to RNA recognition motif, nuclear export signal, and C-terminal domain. And these pathogenic mutations are often found in this C-terminal glycine-rich domain. So in normal conditions, TDP-43 circulates between the nucleus and cytoplasm, and in both, so in nucleus and in cytoplasm, perform different important functions. So it's involved in RNA metabolism, regulates microRNA biogenesis, precipitates in the formation of stress granules, P-bodies, mRNA transport granules. So I think it's fair to say that TDP-43 is like an angel in heaven in physiological conditions. But what happened in disease? So in disease, irreversible shift from nucleus to cytoplasm occurs. And cytoplasmic mislocalization and aggregation of TDP-43 is actually a hallmark of ALS. And just to point out, approximately 95% of ALS cases and 50% of FTD cases is TDP-43 positive. So now we can say actually that TDP-43, because in disease it actually is cytotoxic, becomes fallen angel and really push patients through hell. Okay, and here is a question that raises dilemmas about these TDP-43 aggregates, and this is whether these TDP-43 aggregates cause loss of function, gain of function, or maybe both. And also, here's a question about TDP-43 interactome. Is this interactome the same in disease conditions, or it changes? And when we are examining this interactome of proteins, we have different methods at our disposal. And one of them is also biotin identification method. So in this method, you can pretend like you have biobricks. You take BioID, which is biotin ligase, and you put it together with your protein of interest, which is in our case TDP-43. You then express this fusion protein in cells, add biotin, and when you add biotin, biotin ligase is activated and starts labeling the proteins in the near proximity of TDP-43. You then pull down the proteins and send this to the mass spectrometry, and voila, here is your interactome of your protein of interest. The advantage of this BioID method is also that you can detect transient and big interactors. And this brings us to the core of our mission, which was to investigate the interaction of wild-type TDP-43 and TDP-43 lacking nuclear localization signal, which mimics disease conditions. So, as I already said, so we performed BioID method, and we pulled down the proteins and sent these proteins to mass spec, and we got this extensive list of proteins. And just to make some sense of this data, we first performed bioinformatics and gene ontology. And here are the results. So we first examined the cellular localization of these proteins, and we first observed what we already expected, and this was that interaction partners of TDP-43 wild type were part of nuclear complexes, and in case of delta NLS TDP-43, these proteins were part of cytoplasm complexes. We also examined the cellular compartments of these proteins, molecular function, and roles of these proteins in biological processes. And so here we'll take a closer look into cellular compartments. So interaction partners of TDP-43 wild type were parts of paraspeckles, and the interaction partners of delta NLS TDP-43 were part of cytoplasmic stress granules and cytoplasmic processing bodies, or shorter P-bodies. And now we will take a closer look to the proteins. So from this extensive list of proteins, we chose these 6 proteins for further validation. And what we observed was, so NONO, SFPQ, and FUS. So we already expected that these 3 proteins will be interaction partners of wild-type TDP-43. But what we actually observed was that these 3 proteins also, to some extent, interacted with delta NLS TDP-43. So this is really interesting. And the next 3 proteins are MAML, Pumilio-1, and Etaxin-2-like proteins. And MAML1 is actually, we detected this protein as a unique interactor of TDP-43 wild type. And this protein was actually never linked to ALS or TDP-43 wild type or delta or whatever before. And these 2 proteins, Pumilio and Etaxin-2-like proteins, are actually unique interactors of delta NLS TDP-43, which means that these 2 proteins are actually like gained interactors in disease. We also performed PLA analysis to support our findings, and so you can see the specificity and localization of individual interactions. And this brings me to the conclusions. So protein interactors in the presence and absence of NLS resulted in a list of common and unique TDP-43 interacting proteins. TDP-43 wild type interacts with paraspeckle proteins, but as well also delta NLS TDP-43 interact with these proteins, but just to some extent. And delta NLS TDP-43 interacts with proteins that are part of cytoplasmic stress granules and P-bodies, and these 6 proteins were, so we were the most interested in this. So, and at the end, I would just like to thank my supervisor, Boris Rogel, to send me on this mission, colleagues in the lab, and so also to our collaborators. Thank you.